Thank you all for being here tonight. It's really exciting to be here for what I think has become a wonderful community event. Every year now we're doing this, and it's so in keeping with the Night Campus mission to better connect the amazing research going on at the University of Oregon with our community so people know what's going on, and to partner better with our medical and industry experts so that we can better translate what's going on at the university into technologies that help people. And that's, as the president mentioned, that's what we call the impact cycle. And I'm going to tell you some stories tonight about how we've been successfully able to get around that impact cycle and move some technologies into patient care. But before I do that, I'd like to introduce my family, at least the part of my family that moved with me here to Eugene. This is my wife, Tina, who I met in an engineering class at the University of Michigan, yes, 30 plus years ago. We have two college-age kids who I'll introduce to you a little bit later on in the story. But I think the simplest way I can explain what an amazing opportunity we felt this was that we literally abandoned our two children in Georgia because they're both in college at Georgia Tech. This is Panda here on the bottom. As you can see, Panda loves Oregon, as I think all dogs must. And the picture on the right from a few weeks ago of Panda and I at Spencer's Butte is now part of the standard package we send out to faculty and students that we're trying to recruit to Eugene to let them know, as all of you know, that it's always sunny in Eugene. All right, so in science, we often take inspiration from observations in nature. And so, can we regrow or regenerate organs and limbs? That's sort of the holy grail of the field of regenerative medicine. And the answer is that you can if you're a newt, or a few other special organisms. Unfortunately, you can't if you're a human. And if we look at how the newt is doing this, it's actually really fascinating, right? That a newt within 20 days is able to completely regenerate a lost limb, down to everything's perfectly organized. The bone, the skin, the cartilage are all exactly organized the same way that they were before. And so that raises a lot of really interesting questions. How does that happen? What are the cells involved in making that happen? How do they know what tissues to make and where? And why can a newt do it and we can't? Right? Lots of great questions. And these are questions that biologists are often thinking about and trying to answer in their research. But sometimes the biologists in a place, say, like the Night Campus, might be working next to a bioengineer, like myself, interested in regenerative medicine, and perhaps we have a conversation with a clinician, and we all get together and say, okay, now that we know that this is not science fiction, that this can really happen, how can we use that information to help patients? Maybe we can't completely regenerate a limb right now, but maybe we can regenerate parts of that limb. Maybe we can regenerate the bone, or the cartilage, or the blood vessels, or the nerves. And if we could do that, maybe we could help get away from using metals and plastics that have a limited lifetime, or perhaps a prosthetic. I don't know if we'll ever be able to regenerate like a newt does, but there's advances going on in biomaterials, 3D printing, stem cells, molecular biology that are making their way into patients, and I'm going to tell you about some of those stories tonight. All right, so one great example is cell therapies, and you've heard a lot about cell therapies. There are cell therapies being developed for just about every unmet clinical need that you can think about. There's a lot of clinical trials going on. I'll show you later clinical trials across the entire world and how many there are. A great example of this, and not all those clinical trials are working. And in fact, most of them probably will not work. You shouldn't expect them to work because it's very rigorous to get through the clinical trial process. But there are some that are working, and we're coming up with cures now for things that previously seemed uncurable. A great example of this is cancer immunotherapies. So cancer immunotherapies are basically when you take a patient's T cells and they're being trained, it's a patient-specific technology, the T cells are being trained to recognize their cancer and being implanted back into the patients. When you think about the history of cancer and all the effort and money and time that's gone into cures, it's absolutely amazing that we're living in a time now when there are cures being developed, at least for some forms of cancers. There are still problems associated with this. The blood cancers are much easier to attack than solid tumors. Some patients don't respond well to the treatment, and the cost of these treatments are absolutely exorbitant, up to $1 million a patient. And that's one thing that before I left Georgia Tech, we had started a national consortium in cell manufacturing that now the University of Oregon is part of to address that problem, to develop manufacturing technologies to bring down the costs of these therapies. Another area of cell therapies that has been in the news recently, and you can see the dates here are very recent, is for macular degeneration. How many people in the audience have macular degeneration or know somebody that has had macular degeneration? So these are very common problems that are very difficult to solve, but cell therapies are offering a lot of promise. You may have also noticed some, on the flip side, negative stories in the news that raise concerns about fraud, about potentially unproven or even unsafe therapies, such as the clinic a few years ago that injected stem cells in patients' eyes and blinded them, to medical devices that do more harm than good, and even to the level of very severe ethical breaches, such as the reports out of China of gene-edited babies. Even though these are very rare and outlier situations, they understandably cause a lot of concern in the public, distrust, if you will, and even cause some people to think, well, maybe there isn't value in doing science. But in fact, in all of these cases, it was the science that was missing. And the vast majority of entrepreneurs and scientists and engineers and clinicians that went into the biomedical field did so not for fame and fortune, which unfortunately, to our regret, is very rare, but they did it to help people. And so these are the best way to avoid these situations is really to have rigorous science done that's under strong ethical guidelines. And one of the hallmarks of doing ethical research is open, transparent disclosure of your potential conflicts of interest. So I'm showing here the companies and foundations that we currently work with, as some of whom we've worked with to form ventures that have actually been able to move some of our technologies out of the lab and into people, and I'll talk about some of those. On the right is another example of why it's really important for academics not to be isolated and actually to be working with industry. And that is because business is now supporting a larger amount of their research and development in the United States as federal funding levels have continued to decline. All right, so I'm going to talk about both medical devices and regenerative medicine. The first half will be on advances in medical devices, and the second half will be on regenerative medicine. So I'm showing here some traditional medical devices that some of you may actually have, very common hip implants and knee implants. These were designed originally to be biocompatible. In other words, to not have a negative reaction in the body. They are intended to have they do have a limited lifetime. So if you have one of these, you probably had a conversation with your clinician that went like this. I know you're in pain. I want you to put up with it as long as you can because we don't want to do surgery before we have to, because these tend to last maybe about 15 years before they'll have to be replaced. They're also not customized to the patient. Other than being available in a few different sizes, they're not customized to the anatomy of the patient. This is a rather old hip implant design, but it illustrates that our implants actually do respond in the body, and you get reactions such as at the base of the stem, and this is down in the bone, you can see there's a densification of the bone, whereas up underneath the implant, you can see that there's bone that's been taken away. And so it turns out that bones, just like muscle, actually respond to exercise by getting stronger and to not being used by getting weaker. And so you can imagine as the bone underneath this implant is shielded and goes away, this implant's likely going to loosen and have to be replaced. So the new generation of medical devices, in contrast, are actually designed with a better knowledge of the biology to interact and cooperate with the body and integrate in ways that give you better performance. They're also designed increasingly to be adapted and customized to the patient through, for example, 3D printing, and I'll show you some examples of that. And then finally, they're sometimes designed with materials that actually are intended to go away, so not like metal, which is intended to be there for as long as it'll last, but actually a material that's intended to resorb away and allow the natural tissue to grow. So imagine for this implant, for example, if that implant surface was made of a material that would slowly degrade and allow the cartilage to reform, and the patient could have a living cartilage natural joint again. Wouldn't that be a much better solution? And that's really regenerative medicine. All right, so I'm going to start with a story that's a very personal story, and it's actually one that even many years later is still a difficult one to tell. But it has a good outcome, and it resulted in a medical device that is helping people. So this is our daughter, Sophia, who at age 14 was a nationally competitive tennis player. And she played a tournament and came home and said, you know, my back's hurting a little bit. Not a lot, but a little bit. And so she went in and she had some rehabilitation and she played some more, and then she felt some pain going down her leg. And we said, oh, so we need to get this thoroughly checked out. And the doctors did an x-ray and discovered to our shock that she had a degenerative spine condition in which one of her vertebrae was actually slipped almost 80% off of another. So she had a very unstable spine that she probably had since birth and somehow had adapted to. And so it wasn't discovered until very late. And the doctors told us there was no choice, that she needed to have surgery in order to stabilize her spine. And so Sophia went through a 7-hour surgery to fuse 2 levels of her spine in her lower back. You can see here her radiograph. And what they basically did is they put in screws and rods and stimulated the bone to form between 2 adjacent vertebral segments. And the idea is to stabilize it and remove her pain. And it was a scary experience, I'll tell you. And she came out of it and was in a lot of pain. It was a difficult recovery. But she did everything the doctors said. She worked very hard. She went into high school with what she called her turtle shell and a brace for her back. But she was very brave and did a great job, got back to full recovery. About 9 months later, she turned over in bed and she felt the rods in her back break. What had happened was the fusion part of this, the biology part of it, had failed. And the rods and screws are not intended to work without the biology working. And so we had the second shock of her having to go through a second 7-hour surgery. Now, fortunately, the second surgery went much better. Her recovery was much, much better. She bounced back much more quickly. She never did compete again at singles tennis, but she made her way back in doubles and won 2 state championships in doubles after that. And there's Sophia now. She just is graduating next month from undergraduate school at Georgia Tech, and she's committed to go to graduate school at UCSF. So a great story, ending story for Sophia. But that really led a colleague of mine and I to think about, isn't there a better way of doing this so that patients don't have to go through multiple surgeries like this? And so it turns out this is a very common procedure. It's not common in children like my daughter, but it's very common, particularly in older adults, about 400,000 spine fusions per year. And the idea behind the spine fusion is, again, to stabilize the implant and to restore the disc height. Sophia was actually 1.5 inches taller coming out of her first surgery, which that was the one positive thing about it. In heels, she's taller than her dad. But it also was to remove pain and decompress the pinched nerves. And so one of the ways of doing this is to implant what's called a spine fusion cage. And that cage can be made of metals. Metals have some positive and negative aspects. Or it can be made of a really strong plastic called PEEK. About half the cages are made of this PEEK material. And what physicians like about PEEK is that it can do an image of the patient and actually verify that fusion has occurred. But unfortunately, PEEK also has some disadvantages, and that is that bone doesn't like to grow up right up next to it. And so you can see that here, the bone on the top is in blue, the PEEK, the strong plastic is in brown, and in between forms this orangey stuff that is a soft fibrous layer of tissue. And so what happens a lot of times is that implant will loosen and come out and migrate. So we set out to design a better spine fusion cage using what the doctors like to use, which is this really strong plastic. And the idea was to make it integrate better. And so we thought, well, how can we do that? And we basically took this PEEK material and we designed into it a surface porous layer, about 500 microns, or about 1/2 millimeter in thickness. And when we did that, and we did all the research behind it, you can see here we published 10 peer-reviewed papers on this technology along the way. We found that instead of getting this orangey stuff in between the implant and the bone, all of a sudden we could see the bone growing into all these pores that we had created. And it more than tripled the integration strength for the implant. And then we realized, well, okay, this is great science, but the only way we're going to get this into people is if we form a company and we start working with clinicians to see if they like it. And so we did that. We formed a company called Vertera Spine, and we began working with clinicians, and we began working with the FDA, because the FDA makes you go through rigorous testing to show that your devices are both safe and efficacious. And so we did that, and we received approval from the FDA for the upper spine, which is the easier application, in October, and did our first implantation in March 2016. We then continued to work on the technology and cleared now the lower back, which was the harder one to clear through the FDA. And at that point, two very large medical device companies, NuVasive and Medtronic, came and started competing for our little startup company, which was a nice thing to happen. And NuVasive ended up acquiring it. And that was a great outcome for us, but it also was a great outcome for patients, because now they were able to expand this, and it's now going into tens of thousands of patients. So if you think about what we went through to do this, and we went through basically a typical innovation cycle to make this medical device. It started with an unmet need, with not having a good spine fusion device. We then had an idea to improve it. We did a lot of science behind it. We began working with the FDA to clear products. Once those were cleared and there was a company, you have to go through the expense of building an inventory. Then you have to develop a sales network, and then work with hospitals and surgeons and patients and so forth. This is a long, long process. On average, it takes over 10 years and $30 million to bring a new medical device to the market. And so you can imagine there's a number of good technologies that are discovered in labs that actually never make it into the marketplace because of the timeline and the cost associated with doing this. So the problems arising from this innovation structure are that the design cycle is expensive and takes a long time. I showed it for a medical device. If you're talking about a new drug, it's actually longer than that and probably 10 times the cost, so hundreds of millions of dollars. The implants are also not tailored to the individual, because when you're working with the FDA, this is the way the process works. You have to fix the design and work with that design. You can't change it as you're going through all your testing. So that's a 10-year period, and you can imagine there's new improvements going on during that 10 years. You can't change that design. You've got to stick with the original design. And building up this implant inventory is costly and inefficient. And so an emerging technology for making medical devices is 3D printing. And 3D printing, I'm sure you've heard about it, really started as a prototyping technology where you could make a prototype of something before you actually built it. And then it actually moved into surgical planning. So surgeons would use this to 3D print something they intended to do in the body, and that's still done. It's a very useful application. But it's now moved to the point that we can use 3D printing to actually make devices out of medical-grade materials that have enough good mechanical properties that they can be implanted into patients. And they will address all of these problems. So let me give you a couple examples. This is a clinical example where you've got an airway that's obstructed. The airway can be obstructed because of growth of something on the inside or potentially something that's on the outside of the airway. It can happen in adults. I'll also show you an example where this actually commonly happens in newborn infants and can be very life-threatening. The current solutions for this are medical devices that come in a range of sizes and shapes, but you can imagine there's a great variability in the anatomy of people's airways, right? And so it's never a very good match. And because of that, you have problems with migration, irritation, and plugging. So here's a 3D printing solution for airway implants. And it's going to start out at real-time speed, and then it's going to speed up. And this is made out of a medical-grade plastic material, so this is something that's an FDA-approved material. The process is called fused deposition modeling. And there you can see the implant that's been made. And you can see it has excellent mechanical properties, and it can be handled quite well by the surgeon. So let's think about this process. How did this happen? We had a patient image, so a CT image of the patient. There was then a design meeting with the clinician and a computer model that was made, in this case, of a stent, which means it was on the inside of the airway. The Stents were then printed. They were printed in a couple different ways in case the surgeon had a preference for having little nodules on it or a smooth surface, and then it was delivered for surgery. So how long do you think this process took? 4 days. 4 days from the initial time that the image came in all the way through delivery for surgery. So not 10 years, 4 days. And it's customized to the patient. Here's another example that's even more intriguing, and this was really the first example. This was not, the previous example was a new company that I'm part of called Restore 3D. This was work done by Dr. Scott Hollister, who was actually one of my advisors at the University of Michigan. And then I had the chance to recruit him down to Georgia Tech many years later. So when Scott was at Michigan, though, he was working with a clinician called Dr. Glenn Green. And Dr. Green had a patient who was born with a collapsed airway, a baby, and was on a ventilator. And in the very severe cases, these babies will die. There's just nothing that they can do. They can't leave them on the ventilator indefinitely, and there's nothing they can do to correct it. So Dr. Green had a patient like this. He called Scott, and he said, "Is there anything you can do?" So Scott said, "Well, let me see what I can come up with." And he designed the implant that you can see up there, which is now called a splint because it's on the outside, because the baby's airway is too small to do a stent. And he designed it to be C-shaped so it could grow with the child and out of a material that would go away over time. And so that implant was then printed, and there was some testing done. This had never been done before, so they contacted the FDA, and the FDA said, "This is a humanitarian exemption because this baby's going to die." And in one day, the FDA cleared it to go ahead and do an implantation. So this is Kaiba, the first child that received this treatment in 2012. And you can see the pre-op image here with the collapsed airway. And there's the post-op image showing Kaiba's open airway. And there's Kaiba on his second birthday enjoying cake. And notice this is 2012. So Kaiba's now 7 years old. That material was designed to be in the body for 2 or 3 years, so it's now completely gone, and Kaiba's own tissues have taken over, and the airway is kept open. Okay, so just an incredible story. This has now been applied to babies all over the world. My friend Scott got a call from Saudi Arabia, and he had to fly in and treat a baby there. So it's going through a clinical trial now. Great example, and again, very quickly done. I'll give you one more example in 3D printing because this is a different material. This is made out of metal now. So you can imagine in foot fractures or foot deformities, it's very complex bones, a large number of bones. You can have complex fractures. The current solutions involve taking bone from your hip, which if you've ever had that done is very painful, and transplanting it, or non-customized implant devices like those shown here. And so again, they don't fit perfectly, and they're prone to failure. So here's an example, and this is back to Restore 3D, the company that's based in North Carolina, where we have a complex foot fracture, and we designed for that a wedge implant that you can see has a lot of complexity to it. You can see the outside surface is complex, fitting the geometry of the foot, and we've also designed into it an interior surface that has porosity sort of similar to the spine implant that I showed you. Again, the time frame for this happened just last fall. September 14th was a design meeting. A week later, it was printed, and 2 weeks later, it was implanted for surgery, so a 3-week time period for this innovation cycle. So this is, I think, the future. We're going to see these types of this type of technology be applied in all kinds of different implant areas. You'll actually see 3D printing, I think, in all kinds of markets, and as it continues to evolve and continues to come down in cost. I also just want to emphasize that there's a whole lot of basic science research that goes into this that you don't see. You know, you see when we're the end of it where we're putting it into patients, but there's a lot of basic science research to figure out, well, how do we design this architecture so that bone will grow into it most optimally? And how do we make the interior of it so that it has good mechanical properties and won't fail in the body? And even as you zoom in and you look at the surface of it, it turns out the texture and the topography of the surface make a big difference in whether the cells are going to adhere and actually make bone. And so there's a tremendous amount of science that goes into it before it actually even gets to the FDA. And what that allows us to do is to really customize other aspects of it. It's not just the shape. You can imagine if you have a young athlete and they've got very dense, strong bone, that's a very different implant that you need for an older osteoporotic patient. And so we can design now and manufacture porosities that are appropriate for the patient. And anything that ranges from very strong, dense bone down to thinner bone that's less strong. And just the last slide on 3D printing, because I love this one. The technology for this is just advancing so incredibly fast, and there's many different types of 3D printing now. This is a pencil, and we're zooming in on the tip of this pencil. And as you get in closer, you can see that there's been a castle that was 3D printed on the tip of this pencil. Just incredible resolution. All right, so let's transition now to talk about regenerative medicine. And we already talked about regenerative medicine a little bit because I've talked about the materials that resorb and go away and allow the tissues to form. That's one aspect of regenerative medicine. But more broadly, what we're talking about here is combining cells and biomaterials and biological signals to try and restore function within the body in a biological way rather than putting in a synthetic material. What's shown here in this sort of circular thing is what the original paradigm was for regenerative medicine, where we would take cells from the patient and we would combine those with a biomaterial, maybe incubate them in something called a bioreactor, and then implant them back into the patient. In reality, this hasn't happened very often. In a few rare cases, this has happened successfully, but it's a very complex approach that it turns out probably is not entirely necessary. What's been much more common is for taking cells, either from the patient or in some cases from other patients, and implanting them directly, or using the materials themselves to be implanted and to cooperate with our own endogenous reparative capacity. And I'll talk about that a little bit more. And that's been much more likely to get into clinical use. So over the last decade, I've had the honor of being involved in this field in a very special way. And that is through a Department of Defense funded effort called the Armed Forces Institute for Regenerative Medicine. And this is a very large national consortium, 60 projects, 37 institutions involved. 18 clinical trials have come out of this work. And so there's been a huge impact for our wounded warriors, who you can imagine have severe injuries at a much higher rate than the standard civilian population. My role has been to lead one of the 5 clinical areas. So the particular clinical area that I co-lead, actually with a professor up at OHSU, is the extremity regeneration area. So we work on the arms and the legs. And it turns out that's the most commonly injured tissues in soldiers because their heads and their bodies are really well protected, and that means that their limbs get injured badly. So we have projects focused on regenerating bone and muscle and cartilage, nerves, and blood vessels. And you can see the types of injuries that the soldiers can have, usually from explosive devices. And some of the statistics from this are extremely daunting. Only about 20% of these individuals will get back to full active service duty. And the one that always gets me is that 1 out of 7 of the wounded warriors that are wounded like this will opt for a late-stage amputation. And that means that after a period of maybe 1 to 2 years after they've been through many painful procedures, they decide to have an amputation because they're going to get more function out of a prosthetic than we've been able to give them through our treatments. And so we need to do better for these individuals. So this is an example. This is some of my own work where we're working on repairing function to really large bone defects that won't heal on their own. So this is a typical type of injury that you might see. I've avoided the gory, bloody images, so I'm just showing you radiographs. But this is a complex fracture to the lower limb where there's been damage to the muscle, and it's an open fracture. That means that there's a high probability of infection. And so what the surgeon would typically do in this case is implant a material that's loaded with antibiotic to help ward off the infection. They'll then, at a short time later, 2 weeks later, take that out and implant a material that's intended to maintain the space of the injury, stabilize it. You can see there's a lot of instrumentation that's in there as well. And then that spacer is removed at 6 weeks, and the clinician will typically take bone graft, again from the hip, transplant it down, and then hope that it heals. And you can see in this individual, even though it's a young 24-year-old male, 6 months out, the injury didn't heal, and there had to be a revision surgery. And so we set out to develop a technology to repair bone in these really complex cases more consistently. And here's what we came up with, and we've tried a lot of different approaches. This is our most successful approach. If you have a big injury like this in the bone, it normally would go on like this, and this is called a non-union, so it has not healed. What we developed was a nanotechnology where we would spin nanofibers, sort of like a spider, onto a surface using an electrical gradient, and then form that into a tube. And the nanofibers the cells like to attach to. And the tube gave us an area that was defined by the injury that we could inject into a hydrogel. A hydrogel is a material that's made mostly of water, but also some other components. And that was used to deliver a very potent protein called BMP2 that attracts cells, attracts blood vessels, and actually instructs them to make bone. And so when we do that, you can see here the blood vessels growing into the defect. This is an imaging technology that we developed in our lab a few years ago. And if you look at the healing, we can achieve complete healing of these massive bone defects. And if we take them out and test how strong they are, they're just as strong as actually the original intact bone. So this is in animal testing right now, and we're moving it towards the clinic. We're also working a lot on trying to be able to predict which patients are going to heal well and which ones are not, because it turns out there's a lot of variability in the patient responses. So at 2 weeks, you might see a radiograph like you see here, and one of them goes on to heal wonderfully, and another goes on and doesn't heal at all. And so we'd like to be able to know which patients are going to heal and which ones are not. And so for that, what we've been doing is collecting blood early on. We can collect blood at 1 week and measure a whole host of proteins in different cells. It turns out the immune cells play a big role in this, and develop predictive models where we can predict how well, at 1 week, how well we're going to see healing at 12 weeks. And the idea would be to give sort of the orthopedic surgeon a weather report. And what we'd like to be able to give is a Eugene summer weather report and not a Eugene winter weather report. But even if it is a winter weather report, that's really important. If the clinician can know at 1 week that this patient is likely to go on and not have a good healing outcome, they can change their strategy up front and be much more aggressive. And so this is another technology that I think will be translated actually much quicker than some of the others that we're working on. Okay, so I've mentioned a couple times regenerative capacity. One of the reasons that we see differences between patients is because we have differences in our regenerative capacity. None of us are as good as newts, but some of us are better than others. And it turns out, as you might expect, when we're younger, our regenerative capacity is better than when we're older. When we're younger, we've got lots of stem cells, relatively speaking, and those stem cells produce a lot of factors that are pro-healing. As we age, the number of stem cells that we have declines, and they're less functional. But it turns out that we can influence our regenerative capacity through lifestyle choices. So smokers, for example, have fewer circulating stem cells, and if you exercise, you will have more. And the good news is that if you change lifestyle choices, you can affect your regenerative capacity. So what are stem cells? You know, even with improving lifestyle choices, you may need some cell therapies. And so that's where stem cells come in. Really, stem cells can be defined by two definitions. One is that they can make more of themselves, so they can replicate, and stem cells make more stem cells. The other is that they can differentiate and make different types of tissues in the body. And so the different types of stem cells that you hear about, like embryonic stem cells or adult stem cells, differ in how many of themselves they can make and how many pathways they can form as they differentiate. That's the term we use. They differentiate into nerve cells and muscle cells and bone cells. So an embryonic stem cell can make any cell in the body, whereas the cells that are in all of us that commonly reside on the surface of blood vessels, and you can see them here, have a limited capacity to only make certain types of tissues. So the way I often think about this is when you were young, and this is our son Michael when he was young, you could be anything you wanted in the world, right? Michael wanted to be, I can't remember, it was either a brain surgeon or a neuroscientist and a Major League Baseball player. Which one was it? Brain surgeon. I think it was a brain surgeon and a baseball player. Now, he's studying engineering now in college, so it's pretty unlikely he's going to be a brain surgeon. He's a pretty terrific college baseball player, though, so maybe the Major League Baseball thing is still possible. So that's how I like to think about this as you're going through and differentiating. It turns out we've learned now over the years that these pathways are not completely irreversible. And it turns out the cells can actually jump between pathways. It'd be sort of like you and I changing careers or taking on a second career. And then most excitingly, in 2006, it was discovered that we could take plain old adult cells, just your skin cells or any cell, and reprogram it to turn it back in time biologically into what was very similar to an embryonic stem cell. Have you noticed the embryonic stem cell debate has largely disappeared? The reason is science. So in 2006, that discovery basically removed the need, for the most part, of even needing embryonic stem cells. And I'll show you an example of that at the end of how it's being used. So there's stem cell trials going on all the time. I just looked the other day. I did this search on the 18th, and there was over 5,000 stem cell clinical trials around the world, about half of them in the United States. As I said, for all kinds of different clinical trials, or all kinds of different unmet clinical needs. And one of the main challenges that I mentioned earlier was the cost of these therapies. Basically, in making cell therapies, we are where we were with making automobiles back in the early 1900s. Everything is one-off, and everybody has their own little protocol, and it's not very efficient. And so right before I left Georgia Tech, we formed a national consortium funded by the National Science Foundation focused on cell manufacturing technologies. And now you can see there's a link to the University of Oregon, where we're working on that here as well. And the idea is to bring down the cost of these cell therapies to the point that they're available to everybody, because of course you'd spend $1 million to cure a child with cancer, but that's not something that's sustainable for our healthcare system. And the hope is that we'll see something similar for cell manufacturing that we saw for the cost of the human genome. You may recall back in 2001, the cost to sequence the human genome was hugely expensive, $100 million. And with investment from the federal government, that cost came down over the first 10 years, about an order of magnitude to 10 million. And then there became to be a commercially viable marketplace and investment from industry. You can see how quickly it then declined. And over just the course of 16 years, it went from the cost of $100 million per patient to about $800. So I want to finish with just one last example that I think is really cool, and it's just to give you the idea that cell therapies don't necessarily have to be something that's implanted into the patient, okay? This is work from 2004 using embryonic stem cells from discarded embryos. It's not my work, it was from someone at Stanford, in which they were very excited because they showed they could take these stem cells that have the potential to become any different type of tissue and give them the signals to make them into beating heart cells. They couldn't do it very efficiently. There was only about 5% efficiency, but you can see that it's beating a little bit. So fast forward now to 2019, and this is work by a friend of mine, Dr. Joseph Wu at Stanford, and now you can see there's 90% efficiency, and these are no longer being done from embryonic stem cells. They're being done from what's called induced pluripotential cells. And this is what I was telling you about that invention in 2006, where you can take your own cells, any plain old adult cells, and turn them back biologically in time. And so now we've completely removed the ethical debate. And more importantly, well, just as importantly, these are patient-specific. So they can be from the same patient. So what is he using these for? And you can not look at all the details here, but basically, this is taking cells from patients, say from the skin, reprogramming them into induced pluripotential stem cells, making them into these beating hearts, and then basically you have a model in a dish of that patient. And what can that be used for? Well, say you have a heart patient, which you have maybe 6 different drugs the clinician is trying to decide whether to give to the patient. You could do testing of those different drugs, not on an animal model, not on another patient, but actually on a model of the actual patient you intend to treat through this technology, creating the beating heart of their own cells in a dish. So very exciting stuff. All right, so lastly, I just want to spend a couple slides. That's the end of the science, but I want to spend a couple slides on the night campus, because as the president talked about, this is an incredibly exciting initiative, and it's why I'm here in Oregon. The mission is science advancing society, of course, and the idea is through building these world-class research, training, and entrepreneurship facilities that we can dramatically shorten the amount of time between discovery and societal impact. And so some of the things that we're working on are building a spectacular building, which I assume you've seen over on Franklin Boulevard. We finish next year. We're recruiting outstanding scientists and bioengineers, and the president mentioned we've hired our first 3 faculty already. Forming new partnerships across the state, and he mentioned the Oregon Health Sciences University partnerships, engaging with industry more effectively, because as I showed you tonight in several examples, if we don't partner with industry and don't partner with clinicians, A, you don't know what you should be doing, and B, you can't move these things into patients. And finally, promoting a culture of entrepreneurship in Eugene. And if you remember one thing from my talk tonight, I hope you'll remember that I think what we can create in Eugene is very special. We don't have to be doing, we can do cutting-edge research, we can do 3D printing, we can be making devices that go into patients without having some huge industrial complex. And I think Eugene is the perfect place to have such a nimble, innovative ecosystem. And with that sort of intentional vision in mind, my hope is what we'll create is a place where all this innovation is going on, we can educate our students, and they'll have places to work on leading-edge technology right here in Eugene. And that vision is the vision of the Knight campus, and it's why I came to Oregon. So with that, I'll finish with this quote from Leonardo da Vinci that I really like, which is, there's an urgency in doing, that knowing and being willing is not enough. We have to translate, we have to do in order to move the technologies from the labs into people and impact lives. And so with that, I'll end my talk. I thank you for being here tonight. I hope it was interesting. And on behalf of my wife, Tina, and I'd like to say just thank you to all of you for making us feel so welcome in our new community. Thank you.
Thanks so much, Bob. So bear with us for about 15 seconds. We have a crack team that's going to flip the stage here, and then we'll convert over to a more conversational setting here. So, all right, let's get going with the evening. So, Bob, thank you so much for a brilliant talk and very exciting. It's unbelievable, I think, where we are in this field. And I'll just say personally, it's great to have you here. And, you know, when we hired Bob, we were hoping for the best, and it's more than the best. And part of the reason why, you know, we're not as expert in Bob's field as obviously he is. And I've now given talks around the country, and literally every single place I go, they say, "Oh, you hired Bob Guldberg. Wow, good work." So, so it's terrific. So I'm going to be looking at some of these messages, but just to get us kicked off, you know, it was a bit of a leap of faith to come here to Eugene and the Eugene-Springfield area, I should say. The mayor of Springfield is here, so thank you for coming. So, how's Oregon?
We love it. Actually, you know, we grew up in Michigan, so Atlanta, we were 22 years in Atlanta, but it was always a little bit hot. And so we would escape to the mountains of North Carolina, which actually, it turns out, are a lot like Oregon. So we're loving it. I think the biggest probably hardship has been not being able to watch our son play baseball. But fortunately, now we can get all these games on TV and go back. So we're loving it. We love the community. We love the lack of traffic. And I don't even mind the rain. So
that's great. So one of the first question that I hit was actually interested in the economic impacts, and you talked a little bit about that at the end. As you know, we're making this big investment. There's a lot of faith by the community, but also the whole state. We're a state institution. What do you think about how this is going to be a catchment for some of that work, and how does it make sense? You haven't been here that long, but Eugene in business sometimes have differences of opinion on what that means. And so what's your perspective on how the university can be more engaged in those activities?
Yeah, I mean, I tried to touch on that a little bit at the end. I don't envision, or at least I hope, Eugene doesn't turn into Atlanta with all the traffic and sort of the industrial complex that it's become. But there's tremendous opportunity, I think, for having incubator space for startup companies. And personally, if I was doing a startup and I wanted to be on the West Coast, this would be the place to be. I mean, the cost of living, the weather, the quality of life here in Eugene is so much better than elsewhere. So I think that's a great opportunity. That's a little bit of an unrealized opportunity. We will have actually incubator space in the Knight campus building, and so people will be able to start their companies right there, engage the students to do this. The way I've done my last 2 startup companies actually is to have students that have been trained early on in entrepreneurship and business, and working with Andrew Nelson from the business school, we're starting to build that into the plans for the Knight campus students. And then the idea is to have them go off and do the companies, because it turns out professors actually make terrible business people. So why not let the students do it?
So I wonder if you could talk a little bit about one of the major themes that you touched on is that you were able to really accelerate the movement cycle. And we have a couple questions that, how'd you get around the FDA in that? That part is not totally clear. How are you able to move things so quickly?
Yeah, so I mean, I guess the first comment on that is you learn as you do this that the FDA is not the enemy. They actually serve an incredibly important function. And I would say our FDA is probably one of the more innovative aspects of our government, in fact, where they're really interested in working with the companies to make this work. And so the first thing you do is you talk very early on with the FDA. They do have a challenge with these new technologies. I remember regenerative medicine when it started. The first company, this is a little bit of an interesting story, so I'll take a minute to tell it. One of the first companies was called Advanced Tissue Sciences out of La Jolla. And they were developing a tissue-engineered skin, so a living skin, basically. And this skin was actually made from an unusual cell source that was a discarded medical waste. It was made from a human foreskin. And you could take the skin cells from a human foreskin and make a football field of living skin from that. And that living skin actually was used for treating diabetic ulcers very successfully. It was used for treating burn victims and removed the pain much more effectively than the current technology. So it was a successful technology. Advanced Tissue Sciences went out of business, though, because the FDA didn't know how to regulate them. They'd never had a product like that. It was, they did drugs and they did devices. They didn't do a combination of material and living cells. And so at the time, the company unfortunately went out of business. Fast forward to today, now the FDA has much more knowledge about this. There's a combination product office, and they help to move things through. 3D printing is exactly at the same stage right now. They don't really know how to deal with this because the FDA is used to dealing with having a design, you fix that design, as I said, and then you take it through. And so the way that, to answer the question directly, the way that we've been working with this is that in the one case you saw with the baby, that was a humanitarian exemption. And so the FDA gave clearance because it was an extreme situation for the patient. There's also an exemption called a customization exemption, which is sort of like a physician writing a prescription for a particular need. And so the devices you saw that were customized devices that we're making for adults are through this customization exemption. If we ever want to scale this up and do a large number of 3D printed devices, then we will still have to get full approval through the FDA.
Great, thanks. So one of the questions is about the relationship between kind of these rapid innovations, especially in some respects you can extrapolate this in the future, redefining what it is to be human with these sorts of things. How do we couple this with our ethical considerations and within the university context, thinking about ethics training? How are you thinking about that in relationship to the Knight campus?
Well, it's extremely important, and so we're already partnering on bringing in speakers in ethics and so forth. There actually is a very strong ethics procedures that all faculty have to follow, but I think it's very important that our students get this training early on. And in a broader context, I think it's important that our students are getting not just technology training, but they're also understanding ethics and how to communicate well, and what is the history of these different technologies, and even business aspects and regulatory issues. So we're trying to build in a lot of these sort of, if you will, non-technical parts of the education early on in the process, so that as they learn the technologies, they have that foundation.
So one of the great aspects of Eugene is that we have a local medical community. Do you see potential for partnering with local groups in terms of testing or even development of these kinds of technologies?
Yeah, absolutely. So, and there's several in the audience tonight that I've had meetings with already, because I've always come from a place that had a connection to the medical community. University of Michigan had that. At Georgia Tech, we didn't have a medical school, but we partnered with Emory University and formed a joint department in biomedical engineering. And we're working on something very similar here, where I anticipate our biomedical engineering program will be a joint program with OHSU and also likely with Oregon State. They've been very excited about partnering with us on that as well. But proximity makes a big difference, because it turns out that clinicians are very busy people, and and scientists are very busy people. And so having them very close to each other and having mechanisms to make it easy to work together is really important. So we're definitely going to be working on that. We'd like to have some programs where we can provide some funding for the local clinicians and scientists to work together. And that's so important. It's particularly important for the students, for them to understand what the needs are and not be working on their science in a vacuum.
So one thing you didn't have a chance to touch on are student impacts, and it's still early days for the night campus, but we have a couple questions that are wondering, what's the connection with existing departments and opportunities for students, as well as what's the vision moving forward?
Well, it is early days. I guess I'll speak to one that's getting off the ground now that I'm excited about. The Honors College and the night campus have been meeting about ways that we can partner together. And so a couple of ways that we're doing that is we've launched a program called the Night Campus Undergraduate Scholars Program, in which undergraduates get a full year experience in a laboratory that's actually paid. So instead of just having a summer experience, this is a real research experience, and gives them an idea of whether research is what they want to do for a career or not. And so the Honors College is actually providing funding for a couple of those students. And then we're also talking about, we have a master's program in the night campus that started actually over 20 years ago at U of O. It's an internship program with the students take classes with intense training, then go off and do an internship, and then typically have a job at the end of it. And so what we're talking about doing is an accelerated program with the Honors College, where the students could come in and get basically a combined bachelor's and master's in an accelerated fashion by working through the Honors College and then coming into the night campus internship program. So I think those are the types of innovative programs we'll be trying to put together.
So we're obviously at the cutting edge here, and so we have a couple of questions that are wondering about how far we can push this. So we've touched on it in previous talks about genome editing, and you mentioned it with the Chinese example. So where's the intersection between regenerative medicine and sort of genomic technologies? We seem to be at a potential intersection here.
Yeah. Well, on the one hand, understanding patient specificity, the genomic technologies are going to really help with that. Whether we want that information or not, I think it was going to have to be a personal decision. But certainly in terms of guiding therapeutic approaches, genomic technologies are going to be very useful for that. In terms of gene editing, it's an extremely powerful and in some ways extremely scary technology that can be used in many, many good ways. Actually, the cancer immunotherapies, there's a subset of the cancer immunotherapies that are now relying on a portion of gene editing that will make that a much more effective approach. But when you get into the realm of gene-edited babies, obviously that's a much scarier situation. I actually saw a talk several years ago on this that I thought was very interesting, which was if you think about a person late in life that has a disease that could be corrected by gene editing, and they give approval to have that treatment. Well, that's probably not bad. That's probably okay, right? And then you start thinking going backwards. Well, what if you had a child that had that disease and you knew it was safe, but they really weren't old enough to be able to give permission to do it? That's a different gray area of ethical consideration. And then you start to get into babies, and if a newborn has a disease, obviously they can't give approval for being treated. And then you start to get even into worse areas, like, well, what if parents want a baby with blue eyes or something like that, or someone wants to make a super soldier? You can think about the whole range of things. In this case, supposedly, they were trying to make the baby more resistant to HIV, is what the supposed purpose was. But I think the scary thing is, in this case also, they don't really know what that gene editing did. They knew one thing about that gene and its purpose in HIV, but that gene could have had so many other different functions, and that's part of what makes gene editing scary.
So I want to read this question verbatim. Speaking as one of the many audience members over 50, when or how can I benefit from some of those dang stem cells back to the time therapies? Help!
Can you read that again?
Yeah. When can I get this in my body, basically?
Yeah.
How can I turn back time?
Yeah. Now, I mean, if you want to, now you can. I think.
When is it a good idea to
sure.
steps out there?
That's a different question. Yeah, so again, this has been an area that has been not entirely regulated. So it turns out that if you're using tissues or cells in what's called a homologous, I don't want to use that word. If you're using them where you take them from one person and put them into another, it used to be that was not regulated by the FDA. Think about bone banking, right? There's banks of bones where you can get a bone graft. And so that was not regulated by the FDA. And so it turned out that stem cells kind of fell into that same category. And what that has led to is a number of clinics popping up with unproven approaches where they're willing to, if you want to pay them $10,000, to inject stem cells in just about everything. And so I would not advise anybody to do that. But there's a lot of great science going on and a lot of clinical trials going on, and the FDA is now cracking down on this. So I think we're moving into a more mature state where the cell therapies that are going to come through the clinical trials will be ones that you want to take advantage of. But I would wait for the science.
Yeah, and do we have to tell people it came from a football field full of foreskin cells?
It was
if
one
you bring
foreskin.
anything home tonight, I hope that's the image that you have. This is a more serious topic because you talked about the relationship with the Honors College, but what's the responsibility or the role of the Knight campus in kind of building equity in society and in academic and educational access? What are you thinking about that? How are you preparing for that?
Yeah, I mean, that's a great question and a big challenge. I think, in biomedical engineering, we're a little bit fortunate. It's the one branch of engineering where there's a high proportion of women, for example, in the field. And I'm not sure exactly why that is. It may be because it's a little more focused on helping people. But it remains a huge challenge. And so we're already, even though we're very early stage, we're already implementing some what we call active recruiting measures where we've announced that if anybody has a diversity candidate that they would like to bring in that we might consider it for a faculty position, we'll bring them in to do a seminar on campus. We're also going to be developing a postdoc program where we provide funding for underrepresented minorities to do postdocs. And then I'm excited about implementing a leadership training program where we'll provide funding. This is something that I benefited from a lot when I was a young faculty member to get some leadership training. And there's, even if we are doing okay in hiring professors, you see a real decline in women in leadership positions. And so why is that, and how can we address that? And so I think, doing programs like this hopefully will help.
So we heard some amazing research. You got companies, you got your lab, you're getting things set up, you've got the night campus started. So what have been the biggest challenges that you've seen just over the last year, and how are you possibly doing this?
Well, I'm not coaching baseball anymore, so that helps. Biggest challenges. It's all good. I really learned, and the reason I came to do this is that I had built something similar to this at Georgia Tech, but I hadn't started it. In fact, I took over sort of midstream and I tripled it in size, but I didn't have the chance to build it from scratch. And so this is an opportunity to do that, and that's what I'm so excited about. What are the challenges? I think, the challenges, probably the biggest challenge is getting the word out and attracting really great students. I would say that's probably the thing that I think about the most. I have no doubt that we're going to be able to recruit fantastic faculty, and we're already doing that. But getting the students to recognize that this is an amazing opportunity and come to Eugene, is a bigger challenge. It takes a longer time, and so I want to accelerate that. I don't want to wait a decade for that to happen. And
of course, you have some help also.
Yes, I have some help also, and I prompted him with this question, and then I totally forgot it. Thank you. So, yeah, when I came into this, I was really very fortunate, and I'm going to take an opportunity to thank him because he was the founding acting director of the night campus and did an incredible job getting things off the ground, including the design work for the initial building, and so that's been just fantastic. But I was also blessed with having an incredible staff in place, and they're the ones that really make things happen. They're the ones that organized tonight's event. So if we could just take a moment and thank them for putting this all together tonight.
So that's a great note. We've come to the end of our evening. I really apologize. There's about 100 questions on here, and I obviously couldn't get to them all, so I beg your patience with that. But there's a great opportunity to thank Bob one more time for a great talk, and